Good evening, everyone. Good evening. I am Lauren Henry, Assistant Vice President for Alumni and Volunteer Experience at the university, and like many of you, a proud alumna of this remarkable institution. Thank you so much for joining us for tonight's program. Last month, as I hope you saw, the university launched the Chicago Minds campaign. To be a Chicago Mind is to be relentless, intrepid, and creative, to ask hard questions, follow them wherever they may lead, and advance ideas that shape the world. Let's take a look.
There's something different about the kind of minds that walk these streets and roam these halls. The kind of minds that love to be kept on their toes. The kind of minds that look at the world in a unique light. The kind of minds that aren't afraid to challenge the status quo, challenge what they've been told, that aren't afraid to challenge each other. And most importantly, the kind of minds that are never afraid to challenge themselves. For they know that behind every great discovery, behind every world-changing breakthrough, there have been hundreds of hypotheses considered, reconsidered, and completely rewritten. The kind of minds that dive deeper, minds that are passionate, resilient, relentless, minds that harness the power of the atom, that expanded our sense of the possible and reshaped entire disciplines. The kind of minds that never switch off, even at 2 a.m. There's still debates to be had, opinions to be discussed, and ideas to be formed. The kind of minds that are always open, always growing, formed here and poised to shape the world. We call these minds Chicago Minds.
Tonight's speakers embody what it means to be a Chicago Mind. In fact, she will recognize a couple of them from the video. Each brings a distinct lens to one of the defining challenges of our time. And you, our alumni, our parents, our friends, are part of that same tradition, curiously and passionately engaged with the ideas that matter most. And with that, it is my honor to introduce tonight's speakers. Dr. Kunle Odunsi is the director of the UChicago Medicine Comprehensive Cancer Center, dean for Oncology in the Biological Sciences Division, and the AbbVie Foundation Distinguished Service Professor of Obstetrics and Gynecology. Dr. Odunsi oversees cancer research, patient care, and education at UChicago Medicine. Dr. Jeff Rathmell is the Virginia and D.K. Ludwig Chair, Professor for Cancer Research and Director of the Ludwig Center for Metastasis Research at UChicago Medicine. As a fellow of the American Association for the Advancement of Science, he is a pioneer in immunometabolism, whose groundbreaking discoveries in T-cell and cancer pathways have unlocked vital new therapeutic targets for tumors and immune-related diseases. And Dr. Soni Smith is the Elwood V. Jensen Professor of Medicine and Section Chief of Hematology Oncology at UChicago Medicine, where she is an acclaimed clinical expert and researcher in Hodgkin and non-Hodgkin lymphomas. An elected member of the American Society of Clinical Oncology Board of Directors with over 200 peer-reviewed publications. She is a distinguished organizational leader, dedicated mentor, and founding member of the International Women in Lymphoma Group. Please join me in welcoming our speakers to the stage.
Good evening. So this is a very exciting moment for us, and we're very honored to be here at this HAPA lecture, really to address one of the key challenges of our time, and that is cancer. We want to address what is going on in cancer research and cancer care at the University of Chicago. Why is this important? Cancer is a formidable challenge that society faces. 1 in 3 to 1 in 4 people will develop cancer in their lifetime. All of us knows somebody, either a family member, a relative, a colleague, or a friend who has been touched by cancer. In fact, roughly 2.1 million people in the United States will be newly diagnosed with cancer in this year alone. And around 600,000 will die from the disease. So it is a major public health problem. But the good news is that we've made tremendous progress. Over the last 30 to 40 years, cancer mortality has declined. In fact, the survival rate for all cancers is now about 70%. Thanks to science, research conducted at institutions such as the University of Chicago. So today we are going to be discussing how the University of Chicago is contributing and addressing this major challenge, and especially to talk about some of the new approaches, new treatments, and some of the new avenues that offer hope for the future. So, as you heard, I have with me two leaders from the University of Chicago Medicine Comprehensive Cancer Center, and I'm going to ask each of them to introduce themselves and tell us what exactly they do, including the focus of their research. I'm going to start from Dr. Rathmell, who has been here for a little just over 1 year. Why don't you tell us, as you introduce yourself, why you chose to come to the University of Chicago? And for Dr. Smith, when you answer the question, can you tell us what has kept you at the University of Chicago for more than 20 years? Dr. Rathmell.
So I'm actually from Iowa originally, so I'm from the Midwest. I grew up in this area, and I've trained around the country, though. I went to California, and then actually came back to the University of Chicago for part of my training, and then moved on through University of Pennsylvania, Duke, over to Vanderbilt, and then just last year back up to here. So one of the reasons to come here, one of the driving reasons to come here really is, for every place I've been, and one of the things that stands out about University of Chicago is it has this incredible drive towards fundamental discovery science. I'm a basic scientist. I'm interested in trying to understand the mechanisms that drive cancer, mechanisms that drive immune responses, with a focus on how metabolism links it all together. But here we had, particularly with the Ben May Department for Cancer Research, where I'm now the chair, a department and a group that's focused on basic discovery, but towards a disease with an application. And there's not that many departments for cancer research out there in the country. So that actually stands out, and the institution, of course, has a remarkable history. And then you put that in the context of the investment that the institution's made in cancer biology and cancer care, and you're going to hear a lot more about that from Kunle Odunsi. It just became kind of a unique opportunity. The other part of it is, there's sort of life events that happen, and my wife, also from Iowa, we've tracked around together. But she was with me. We had a jointly run lab at Vanderbilt where we studied kidney cancer. She went to Washington, D.C. and worked under the Biden administration, and that came to an end, and so she was looking for a new job, and so all of us were sort of up in the air, and this is where I landed, and I'm very happy to be here.
So I'd like to just extend my warm welcome. Thank you all for taking some time out tonight to talk about cancer at the University of Chicago. So as Kunle said, I'm Soni Smith, and I'm Chief of Hematology Oncology, and I came to the University of Chicago in 1998 for my fellowship training, so specialty training in hematology oncology, and joined the faculty in 2001. And most of my career, I've been a clinical investigator. So I've written about a dozen clinical trials, and then, the nature of rare disorders like blood cancers, including some subtypes of lymphomas, have been able to partner with different institutions around the country and around the world to help bring new treatments forward. Throughout this time, I am super excited that we've been the first for several new leaps within the field. So the first person to get CAR T-cell therapy in the state of Illinois was my patient, and doing really well 9 years later. And we've seen a lot of this type of progress. And then just in the last 7 years, I became the chief of hematology oncology, and it's really been a pleasure to be able to recruit some of the best and brightest to our university. Kunle asked, why I stayed. So, I came here for training, but I stayed because, and I'm sure this will come as no surprise, there is such a high level of discourse and dialogue and questioning and curiosity. And to find all of that in a concentrated group of people who are singularly focused on improvement of what happens, for people who are going through cancer or discovering, anything at the bench in the lab that can be taken forward is really exciting. And I sincerely believe that we have all of the ingredients to do almost anything at the University of Chicago. We are all co-located on one campus, and the ability to interact with different disciplines is a real strength.
If I may just add to that, so I came to the University of Chicago only about 5 years ago, so less than 20 years. And for me, I was drawn by the culture of innovation and discovery. Innovation and discovery, which is something that happens every day on that campus. And this is not just about discovery that just sits on the shelf, discovery to impact human lives, which is the culture at the University of Chicago that really drives what we do. I'm going to stay with you, Dr. Smith, really to talk about this new infrastructure facility, the AbbVie Foundation Cancer Pavilion. What exactly is it? How is it going to drive your work in terms of caring for patients as well as research at the University of Chicago?
Thank you. So this is, I think, a really exciting time for the University of Chicago as it relates, in many ways, but certainly as it relates to medicine. So we will be opening the first freestanding cancer pavilion in the state of Illinois, and the only one outside, in the Midwest, outside of the James, actually. That's the only other Midwest freestanding cancer facility, pavilion. And the piece about it that is really exciting is that I think anybody who has experienced going through cancer themselves or for a loved one, you realize that things can be very fragmented. You have to go from one place to another, to another, to another. And what this is going to be is not just a physical space to get everything under one umbrella, but also a transformative culture so that people can come, feel safe, know that their problems will be addressed and hopefully solved. And we've never had this kind of opportunity. And we want this pavilion to be for everyone. So really excited.
Yeah, let me switch to Jeff. So AI is here to stay with us. I'm sure there are many in the audience that are familiar with artificial intelligence in our everyday life. It is here to stay with us. In your own work, how is AI influencing basic translational research that you are doing or your team is doing at the University of Chicago?
Yeah, so AI actually is changing everything amazingly fast. An example for just myself. Okay, so there are AI tools that allow one to analyze all this gene expression data that's been accumulating for now years. When someone does an experiment or a study where they collect gene expression data, that all gets deposited in big banks. It's all accessible. But the problem is the only people who can really get at it are very well-trained bioinformaticians. So it's a very specialized skill. So most of that data is kind of out there and hard to get at. And in our own lab, when we do a study, we put our data there, and we have a bioinformatician who can look at that data. But it's a huge bottleneck because there's not that many people who can do it. So 2 weeks ago, actually, I decided, okay, I'm going to try to see if I can use AI to do some of this, because everybody tells me you can. And so at 4 o'clock on a Saturday afternoon, I downloaded some data just by telling it to download the data. That was the prompt. And by 6 o'clock, I had analyzed the whole thing. And I had never been able to do that before. And it opens up the door for everything that's been done before that is out there in the National Library of Medicine. And what that allows you to do, not just get past this one little bottleneck, but what AI is great at is taking these really complicated data sets and finding associations and coming up with new ideas, because then the other thing it'll do, it'll look back at the other existing published literature and trying to make connections for you and come up with hypotheses. So those hypotheses may or may not be right, but you can then filter them and come up with new things. So then the bottleneck is not, can we do the data analysis? The bottleneck is, can you go back to the lab and now test it? So from that analysis I did at the very first time, we came up with an idea. We had a project where we didn't really know what was happening. And there was one thing that stood out, and I asked one of my students if she could take a look at it, and lo and behold, there it is. It was correct. So everybody in my lab is now switched, and this is the first thing they do when they get to a problem. It completely flips where the choke points are in science by eliminating the challenge of how do you manage this huge amount of data. That's what it's happened in our lab, and it's a simple thing, but it's pretty transformative for our group, and I know other groups are like that. Other aspects of AI, not just analyzing these big bioinformatics types data sets, but drug discovery. There's a big program at Argonne, and I know that you, Kunle, are involved in this, where you can, instead of doing a huge chemical screen to find a new drug to hit a specific target you're interested, now AI can go through and it won't necessarily tell you the drug, but it'll narrow the universe down to something that you can attack very, very easily. It shortens the time frame dramatically. So this is going to speed up the progress tenfold, hundredfold. It's going to be remarkable. I'm super thrilled about it.
And I'm going to ask you the same question, but in a slightly different way. How is AI impacting how we care for cancer patients, whether it's in the realm of prevention, treatment, survivorship? And a second layer to that question, that there's some issues about ethics of AI. Can you speak to that as well?
Yeah. So, on the clinical investigation side, I think, there's a tremendous opportunity on discovery, building on what Jeff was just talking about, and then applying it specifically within our cancer, in our population of people who have cancer. And so one example or one story of that, is that we have a Center for Computational Oncology, and there's actually a BSD-wide and a cancer center huge initiative on understanding the application. So one example is Alex Pearson, for example, who is a specialist in head and neck cancer. Looking at, just with like an iPhone, taking pictures of people's mouths and predicting whether or not there's a head and neck cancer that is going to develop over time. Now, this is, application both here, but also he has partnered with different sites within Africa, to start screening for oral pre-malignant lesions, precancerous lesions of cancers of the head and neck. And that is all using, essentially a digitization of the picture and then being able to predict. And that's just one of these applications. Another person who is in that same center, Fred Howard, is looking at breast cancer biopsy specimens and looking to see if we can predict which mutations are there based on the pathology being digitized. And you can do the same kind of thing with radiomics. So with CT scans, with PET scans, to be able to really look at what's happening in a person's body without having to do a biopsy. So I think the application of AI there, taking those little data points and putting them together into a story that can help with either prediction of cancer, diagnosis, prevention, is really, really important. On a clinical side, I'll tell you, our new hospital is new cancer pavilion is very tech forward, and it includes elements of AI. And so an example of that is that, as we're designing these rooms, of course, there's a lot of tech in terms of, ambient listening, but one of the things I'm super excited about is that we have partnered with a company called Artisight. So some of our beds, for example, if anybody's been admitted to the hospital, you get woken up at midnight and like 2 a.m. and then 4 a.m., right? I mean, it's like you just keep getting woken up. And what the technology here does is it can predict fall risk, for example, by looking, not filming. There's no video involved. There's, so the privacy piece you talked about. It's not that, anybody's personal face or, any other features will be kept. But for example, it can make inferences. Is the person in the bed horizontal? Are they, is their heart rate going up? Is their temperature changing? Is their blood pressure changing? Without somebody having to come in and interrupt, that sleep, which I think is really important. That's just like one example of AI in action. And then the privacy part, is taken very, very seriously. And again, that's where we have made certain, certainly for the clinical side, that personal information is not part of what is collected through our AI technology, that it is more inferences than anything else.
Yeah. So as we think of making progress in cancer research, Jeff, there has been significant funding cuts from many angles. So one question is, how are the funding cuts affecting cancer research, based on your experience? So for example, we used to be funded when you submit a grant proposal to the National Cancer Institute and you score 9th to 10th percentile. Now it's down to 4th percentile, meaning for every 100 grants that is submitted, only 4 of them will be funded. That is a pretty high bar, very tough. So how is that affecting research? How is it affecting early career investigators? How do you think it's going to affect the leadership of the United States in cancer research going into the future?
Well, it's a real problem. Even at the 10% level, it's a challenge. We're actually, if you go by the NIH guidelines, there's policies as to how many grants you can submit. Some of that's in response to AI and people using AI to write grants. But you're allowed to submit 6 grants in a given year. So if only 10% are funded and you can submit 6 a year, it takes you a year and a half to get a grant. And it takes maybe a couple weeks or a month to write a grant. So you can envision how much time people take. And then when you lower the percent down to 4%, it becomes even more of a a little bit of a crapshoot. There's a lot of randomness in the system. So it becomes very hard. We need to find alternative sources of support. That's philanthropic, that's commercialization. There's a lot of foundations, of course, that we can reach out to and people can apply to. It's particularly challenging for the junior faculty, the faculty who are just getting started. It's sort of a daunting task to know that the funding rate is so low. So it is scary for them. One of the things I've seen, though, is if the funding rate is 4% or give or take, as people say, well, why should I even apply because the odds are so poor? The reality is many of those grants that are coming in, there's a lot more grant applications. That's part of the reason the number's low, as well as the funding. Many of those grants are simply because people feel pressure to put out more and more. So good quality still can rise to the top. So I always encourage junior faculty and all the faculty that if you just focus on writing good grants, and there's still chances there. Not as good as we'd like, but they're still there. And just to be entrepreneurial, look around for other opportunities. Now, as far as the global aspects of this, it is true as the United States contribution and investment in research and development has gone down, and as a proportion of the GDP, it has gone down. It's been relatively flat at a dollar level, but it's gone down GDP. Other countries and other areas of the world in essence, going the opposite direction. I guess China is the most obvious example. So the investments in research there are quite strong. The biotech industry in China is growing very fast. And it actually is, it's a huge competitor for us now. When we recruit faculty to a faculty search, it used to be that people would apply, international scholars would apply, and many would want to go back to their home country for a variety of reasons. But the reality was we didn't lose a lot of those candidates. They wanted to stay. That's not always the case anymore. And a lot of that's financial. There's some political aspects of that too. But we are I think we're at a kind of a danger point where we have to make it apparent that it's possible to succeed for the junior faculty, and it's a good job, and it's a good career, and it has a lot of impact, and so there's a lot of value there. And hopefully we'll raise that 4% up. I think the ship is turning a little bit, or at least stabilizing. But yeah, it's a big challenge.
Yeah. Part of the problem also is the fact that there has been some erosion of within the public about the trust in science. So maybe, Soni, if you want to answer this question. So is this a misperception, this erosion of the trust in science? What are your thoughts about this topic?
Yeah, I do think there has been a general erosion is a good word, of how science is perceived in the general population. And some of this is related to the ability to obtain information so quickly, right? Whether it's through an AI search or, the old Google search, I guess I can call that old now. You get a lot of information right away. And, I think a natural tendency for many people is like, if they see it there, then that's a source of truth. And the thing about science and about clinical research, for example, is that there is a burden of proof we have to get over. And that involves rigorously designed, well-conducted, ethical trials to show whether or not a treatment that we say is useful for a particular condition actually works. And of course, we have a lot of oversight from, the government and the FDA, but those trials are really good sources of information, and they are very different than testimonials that can be found very easily. And so I think that's a big part of it, is just, the access to information and the willingness to sort of challenge any assertions that you find online versus what is told to you, in a clinic or in, a scientific journal, for example.
Yeah. So let's begin to talk about some of the new therapies on the horizon. And let me start with you, Jeff. So your work focuses on the interactions between the immune system and cancer, especially something that you call immunometabolism, interactions between metabolism of cancer cells, metabolism of immune cells. So how are we likely to take advantage of this knowledge for new therapies? So for example, vaccines are coming up, that people can be vaccinated to either control cancer or to prevent it from coming back.
Yeah, well, so the last couple of questions were a little doom and gloom. This is, I think, the opposite, because we are at a point right now where the technology allows us to do remarkable things. This is one of the best times to be a scientist from just the capability aspect of it. So the immune metabolism area, we're trying to understand how cells use nutrients, how your body uses nutrients, how that affects an immune response, but cancers as well, and how it affects an immune response against cancer. So vaccines, that's part of it. We actually, in our lab, we have a big focus on obesity in cancer. Obesity is the #2 modifiable risk factor for cancer, behind only smoking. But at the same time, individuals who are obese will often respond better to immunotherapies. So it's this kind of funny paradox, why it's worse and also better. The mechanisms of that we don't really entirely understand, but they become really prevalent because obviously obesity is a big issue. And then you have the other things like the GLP-1 drugs, which are now out and been wildly successful. They seem like they cure just almost everything. And they have big impacts in cancer too, so we're looking at that. So there's a lot of technologies out there trying to understand how your body's metabolism, how the tumor metabolism, the nutrients that it uses affects the nutrients that a T cell, which would kill the cancer cells, what does it need when it goes into the tumor? So in the vaccine setting, we're trying to enhance an immune response, so the T cells, which would kill the tumor cells, so they can get into there, and they're in this space where the cancer cells are using all the nutrients too, and so how do they interact, and how do we make a vaccine, and how do we make a T cell best able to survive that hostile environment? How do we make it resilient to the stresses that are there? And there's ways to do that, ways we can build that up, but we have to start by understanding what the baseline is. And so the kind of tools that we have now to understand within a tumor what actually is happening at a cellular level, thinking about the individual cells, but also the clusters of cells, the groups of cells, the areas, the locations within a tumor, we can now understand genetically what's happening to those cells, metabolically what's happening to those cells, how they're communicating, and that gives us the kind of targets and the ways that we can think about how to build a better vaccine, how to build a better CAR T cell, for example.
So in terms of specific cancer types, Sony, why have we seen dramatic improvements in outcomes in some cancers? Some cancers respond very well to treatment, some do not, such as pancreas or glioblastoma, and so on. So it's always a question that I get asked a lot. Why are some people responding, some people are not? Why are some cancers responding, others are not? And can you give an example of a clinical trial that you are very excited about that is going on at University of Chicago right now or completed that you are very excited about with regards to these cancers?
So, cancer, we use it obviously as an umbrella term, and it hides hundreds, maybe thousands of different diseases that are driven by different mutations, different biologic pathways that are abnormal, and that's just focusing on the cancer cell. As we heard from Jeff, that cancer cell doesn't live in isolation. It lives within an immune microenvironment, which lives in a body that has a microbiome, a gut microbiome, an oral microbiome, a skin microbiome, and is then, subjected to different exposures. And I think that type of, heterogeneity of how cancers survive, the smartness of these cells just cannot be underestimated. It is just so, it's so vast. And so, one of the things at University of Chicago, which I have really appreciated, is the ability to be interdisciplinary, to work with people in the chemistry department, to work with people on the microbiome, to work with people who are developing cell therapy for solid tumors, which Dr. Odunsi is, on the leading edge. There's so many different complexities that we have to consider, and to know that it's all there is really exciting. There's so many different trials we have right now. Perhaps one I could just talk about that is getting close to activation actually is one of your studies, if I may. I think maybe some of you know what CAR T cell therapy is, but I'll just spell it out for a second. So it stands for chimeric antigen receptor T cells. And so the concept is that we take a person's T cells and in the lab use a retrovirus to re-engineer them to now have a special receptor on the surface that looks for the cancer cell protein that you're trying to target. And when it finds that protein, it triggers that T cell to massively divide, and then hopefully do its job, which is to get rid of that cancer cell. And so this type of CAR T cell technology has been around now, and it was first developed in lymphomas, which is how I got involved with it. And it's been a great way to offer opportunities for people who previously would not have survived to now have that long-term survival. But it's limited to certain blood cancers that have proteins you can target. What Dr. Odunsi has done is look at targets in solid tumors, right, which are much more common than blood cancers, ovarian cancer, now even lung cancer, and to find proteins that are differential. And so one of the trials that I'm really excited about is a homegrown trial that is where the CAR T-cell product is actually being engineered in a facility at the University of Chicago. And this is just an example of bench to bedside where a protein is identified in these solid tumors that is unique to that and doesn't have sort of these off-target effects, and then you can develop a T-cell to go back into a person's body, find that protein, expand the T-cell, and kill the cancer. So I'm pretty excited about that.
Good. Well, I'm glad you brought up CAR T cells. One of the questions that actually came in was about the use of a technology called CRISPR technology. Jeff, maybe you want to tell us what is CRISPR and how can it help in improving the effect or the job of CAR T cells? How can it help them?
Yeah, so CRISPR is, it's a long acronym, but essentially this is sort of the poster child for the pure basic science discovery having a massive clinical impact. So this comes from the discovery of how bacteria fight off invading viruses. So bacteria can be infected by viruses the same as we can. So CRISPR is a mechanism that bacteria can take the viral DNA and chop it up to bits. So that approach, though, you can tailor in many different ways to do very precise genomic editing and genomic modifications. So that gets used in the lab. We use it all the time in the lab to do genetic screening, to identify new targets. That can be useful for CRISPR. We have a target or two ourselves that have come from that. Where it really, though, has an impact in the CAR T-cell space is allows you to very precisely do these genetic modifications. Just to make a CAR T-cell, you have to take that chimeric antigen receptor and get it into the T-cell. So you have to insert that DNA into the genome. So you need to do that in a very careful way. Now, the standard way that it's done right now is we use retroviruses, like Sonny mentioned. They will insert the DNA, but kind of it's a little bit random. So you don't have full control over it. But with CRISPR, you can say, I'm going to insert this piece of DNA into a very specific spot that I know exactly what's there. I know it's not going to cause any danger, and it gives us the ability to have a lot more control. So one of the applications of that, outside of just very precise CAR T-cell editing and the kind of engineering that people can do, the synthetic biology, is remarkable. People are incredibly creative about how you can put these pieces together. It's like it's like a Lego set. You can make them however you'd like. So that's all going on. But it also opens the door for in vivo CAR T cells, where you can change the entire manufacturing process by now using CRISPR to insert the DNA into a patient, into the individual, rather than do it in a manufacturing process outside of the patient. You wouldn't do that ordinarily because you wouldn't know where it's going to go, where the DNA is going to go, and that is potential danger. But because you can target it specifically, you can put it exactly where you want it to be, and all of a sudden the safety profile and the efficiency can go way up. So this is a new frontier. It's just starting to happen, but it's going to be probably the future, I think. But CRISPR has opened up the genetic toolkit in many ways.
That's amazing. One question that I got asked by someone yesterday who was sitting in my office is, are we going to see a cure in our lifetime? So let's go to the future. Can you speak to maybe one potential advance in the next 5 to 10 years from where you sit, from where you see things, 5 to 10 years? And maybe I will try and address a cure in our lifetime?
I think the answer is yes. I do. I think for more and more. We've already seen, tremendous advances, but I think between, what we already talked about with AI, being able to understand how cancers outsmart our current therapies, which are blunt instruments, right? Chemotherapy is a blunt tool. So I think understanding what's happening in the cell and then being able to rapidly screen compounds. And this is something where, our partnership with Argonne, I think, is particularly exciting. And so I think we're beginning to crack the code for many cancers that have previously been resistant. And, the University of Chicago was actually part of the clinical trial that some of you may have read about in the newspaper for pancreatic cancer, where there's a new drug against a protein that has previously been considered undruggable. We can't target it because it's too smart. It shapeshifts and whatever this protein does, we can't block it. And now using newer technology, looking at different fragments of this protein, we actually do have a drug that can target it. And it's just the beginning. So I think yes.
Jeff?
Yeah, I have a lot of hope as well. The KRAS example that Sony was mentioning is remarkable. And it's this culmination of a tremendous amount of basic science and clinical work kind of coming together in a really nice way. The part that gets me excited and I think has a huge opportunity is kind of the next phase that emerges from immunotherapy. So, we've got chemotherapy, radiation therapy, precision therapy like the KRAS inhibitors. Immunotherapy has been the hot topic for the last 15 years. And that has been based largely on a very small number of interactions and a very small number of cell types, these T cells and how you can get them to fight a tumor. But I think one of the next phases that emerges out of that is actually the sort of light bulb going off in the recognition that cancer cells are not alone. They're mixed in with many, many other cells. Immune cells are part of that, but they're only part of it. There's a lot of other things going on. There's diet that have a whole body effect. There's sex, there's age, there's many effects that can come back to the cancer cell from the host, from the patient. But then even within the tumor, there's lots of different kinds of cells there. It's not just the T cells. There's many, many cell types that could impact the way a cancer can progress and potentially treat it. So I think there's a whole universe of new targets that we're just starting to understand right now. And that's emerged from the immunotherapy field, but it's just expanded.
Well, let me thank both of you, but also just to make the comment that I think we stand at the edge of an unprecedented scientific promise. This is a moment in history where we're poised to really tackle one of humanity's greatest problems, and that is cancer. At UChicago, there are two things that I'm really very excited about in the coming year at least, and I think the next 4 to 5 years. Number one is the Cancer Pavilion that we talked about. That is going to transform care in Chicagoland. The second thing that I'm excited about is our efforts in therapeutics, new drug discoveries. We are establishing a therapeutics discovery institute. I think, Jeff, you kind of touched on it. This is where we integrate all of the latest technologies, including AI, to rapidly identify drugs for our patients. And this institute has this hybrid model of really translating from bench to bedside, from the laboratory to the clinic very rapidly by engaging with industry, by engaging with commercial partners. It's a collaborative institute that engages all of the UChicago ecosystem, the chemists, the biologists, the clinicians, as well as Argonne National Lab. So we are very excited about what's going to happen in the next few years at University of Chicago and what's going to happen to cancer. I think cancer is going to be cured in our lifetime. So I completely agree. So with that, we have time for Q&A. If there are questions from the audience.
Good evening. My name is Chuck Okuje, licensed architect. My question is, hello, I'll stand up. So hi, my name is Chuck Okuje, licensed architect, lifer at University of Chicago, and I value the education I got there, and as well my family. That said, how do you see science combating the future of the K economy, where the upward slope is corporations and the downward slope is individuals, and where we typically stand as researchers and service providers, educators, is that middle ground? If that's eliminated, how do we hold the line against making sure that cancer and other diseases that affect all members of our population, there's equity and access to those treatments that are applied to all and don't follow the same lines of a K economy up and down?
You want to give it a try?
Oh, sure. I mean, you are speaking to what is at the heart of our cancer initiatives and programs at the University of Chicago. You know, the space that has been created is there for all, and we've made really concerted efforts and, you know, dialogue and interaction to, for example, the South Side, with investment in, you know, people who are living and working there, so that this is a place that, you know, anybody who has cancer can come and know that they will be taken care of. There's just been a tremendous amount of outreach in terms of prevention. You know, we have the Ralph Lauren Center. I don't know if you want to talk about that, Kunle. It's a really, you know, I think wonderful partnership that reflects our core values. I agree with you. I think that things are at risk for being, you know, removed from the people on the ground. And we just have to keep our eye on that and advocate and use all of our powers of advocacy to make sure that that remains a priority.
Yeah, if I may just add to that, at the University of Chicago, we have a lot of programs designed to address that question of making sure that we address the equity gap. Intentional programs, we have a community outreach and engagement, prevention programs, education programs in the community. And one of the latest ones that I'm most excited about is a partnership with a foundation, the Ralph Lauren Foundation, where they provided for us a grant to create a very extensive navigation system. How do we best navigate patients in the community to the best, most appropriate place for them to receive care? And how do we navigate patients within the health system? Once they come in, how do we make sure they are getting the appropriate care? So there's a Ralph Lauren Center that will be in Hyde Park in the new Cancer Pavilion. But importantly, we've actually located one now in Ingalls Memorial Hospital, which serves a population that we really want to reach to make sure they have access to care. So there's a lot of activities going on to address that question.
Hi, so I'm Annie Logue, and I'm Booth class of '91. And you talked about a cure for cancer in our lifetime, maybe. Is glioblastoma included in that?
When we say cure for cancer, we mean all cancers. All cancers. No cancer is left behind. There's tremendous effort going on. I mean, even for nervous system cancers, brain cancers, pediatric tumors, we have working groups. We have a lot of work going on in those areas, including glioblastoma. I just came off a meeting this afternoon with some colleagues, a meeting supported by the NCI, focused on cancer vaccines. Glioblastoma was on that list. And there are already spectacular results of using cancer vaccines in patients with glioblastoma. And there are ongoing, we're talking about even how to improve upon those results. So the answer is yes.
Suzanne Gilbert, Booth, long time ago. Anyhow, at one of Harper lecture a number of years ago, they talked about making drugs and having a person's individual DNA see how they're going to react to drugs. So my question is, with immunotherapy and some of the cancer, but immunotherapy really, is there any way or is there any work being done to know if an individual is going to negatively react to it?
Yeah, you know what you're bringing up is really important because it's not just the effectiveness of the drug, it's also the side effects and the toxicity of the drugs. And, you know, I think that for a long time, you know, when we think about what's called precision therapy, what we're thinking about is a person's DNA, you know, primarily the DNA in the tumor. But I think what we're realizing now is that a person's response to a therapy as well as the side effects to a therapy are shaped not just by the DNA, but also everything around it, including the microbiome and the microenvironment. And you know, one example of that is another trial, which one of our physicians, Dr. Dan Olson, just opened, which looks at the gut microbiome. And you know, well, actually, let me take a step back. Tom Gajewski, other investigators at University of Chicago and elsewhere have shown that the ability to predict response may actually lie in the composition of the bacteria that live in your gut, because that exposure to bacteria trains the T cells. So when you then get immunotherapy, you may have a better or not so good response. And so taking that one step further, the trial that we have right now is actually using a very commonly used drug called lactulose and seeing how that affects the microbiome in response to CAR T cell therapy. So I think what you heard in the past was like the beginning of us understanding how to predict who responds to a specific therapy and its side effects.
Hello, my name is Abel Robles, and I have a question. As a lymphoma survivor, I received chemotherapy and immunotherapy as my initial treatment. Given the promising result we are seeing with CAR T therapy, why can patients start with CAR T from the beginning instead of going through the chemotherapy first?
Yeah, I think you're asking a really important question, and I do think CAR T is being moved forward in lymphoma therapy, but it also, you know, it's a bit of a balance because it's also very involved. I mean, at least right now, the majority of people who go through CAR T, it is, you know, 2 to 3 weeks in the hospital with, you know, a recovery period, whereas some of the other therapies may be a little bit more accessible and well tolerated. And they do work for a good portion of people, just not everybody. So I think CAR T is being moved forward, but we're trying to understand who needs that up front and who might actually have a chance of being cured with the more standard approaches. And in the meantime, we're trying to take the standard approaches and reduce the amount of chemotherapy and increase the number of targeted agents. And a new category of drugs, which we didn't talk about here just yet, is bispecific antibodies. You know, so antibodies that target more than one protein. So I think it's there for some people.
So we have time for one more question.
Thank you. David Huber, college, class of '88. 2 quick questions. First question is, I've been hearing a lot between the correlation between the mRNA COVID shots and dermal cancers. What are you finding in your research? And 2, why the prevalence of pancreatic cancer and what causes that?
Do you want to?
Wow, that's a tough one. The COVID vaccines and cancer, yeah, there's some evidence that if you increase your immune system, you can fight off cancers in a way that doesn't seem entirely intuitive in terms of it's not necessarily cancer specific. What it can do is it can increase inflammation, increase the sort of the tone of the whole immune system, and that can help fight cancers. So there's some evidence with the COVID vaccines that cancer incidences can be changed. It's also some evidence with other viruses. It's not just COVID, not just with the vaccine, but other viruses that if you reactivate the immune response against them, even though it's not the tumor, they can basically everything goes up. Ship lifts all boats, right? And so that might be contributing to that part of it.
I can
Pancreatic
speak-
cancer.
Yeah, I was just going to say, you raise a really, really important question because colorectal cancer is on the rise, pancreatic cancer is on the rise, and affecting younger people. Like it used to be that, the median age at which somebody would be diagnosed would be like 60 or 70, and now it's more like 40 and 50. And why this is happening is a really intense area of investigation. And one of the things we will have in the new Cancer Pavilion will be a center focused primarily on early onset cancers that includes pancreatic and colorectal cancer.
Okay, thank you again, Dr. Rathmell and Dr. Smith. And thank you to the audience for your attention. At this point, I'm going to invite Lauren back on stage.
All right, well, I will first thank you all again and ask you all to join me again in thanking our amazing panel. And of course, thank all of you for being here tonight. Thank you for your incredible questions. I always find myself leaving these conversations both energized, but also with so many more questions, which is a feeling I associate just so deeply with our university. So I hope you're leaving feeling the same and knowing that simply being here tonight, being engaged, asking your questions, staying curious, you are our Chicago Minds in Action. And we're so grateful to you for being part of the community. And with that, I will just say thank you all once again for joining us. Please enjoy dessert, drinks, and more conversation. Thank you.